Today I'm going to get started talking about nerve cells themselves and then the nerve impulses, and so about some basic neural communication. The nervous system is comprised of two basic kinds of cells, neurons and glia. We have about 100 billion individual neurons. Obviously nobody's counted them, but that is the estimate. And I have this really brief history. It's more and more brief every time I do this, is the kind of what I feel like is the necessary background of the physiological approach. And so we're starting by defining the nervous system, because by the 1800s it becomes very clear that in order to really understand human behavior and thought, we need to describe the nervous system. And at this point, there were still two opposing ideas about how this all worked. Reticular theory, those people believed that we had this large network of fused nerve cells. So you can imagine this is kind of like a net of connected cells. And then neuron theory, that the cells were distinct elements that were making up the nervous system. So these neurons were separate and they somehow had to communicate with each other. So as they were trying to describe and define the nervous system, Camille Golgi in 1873 came up with this method of staining brain tissue. So using a solution of silver nitrate, which would allow for some neurons to be stained and all the other neurons to not be stained. And so if you think about how billions of neurons are going to be mushed together, we're not going to be able to tell what that is, right? Well, here you can see a silver nitrate stained neuron, but you can also tell that there's still no way to really note that microscopic gap, those synapses between neurons. So Camille Golgi was one of the people who was a proponent of reticular theory. Actually, he thought these neurons were fused together, and this staining method didn't change that. In the late 1800s, the Spaniard Santiago Ramón y Cajal was the first to demonstrate that the individual cells were what were comprising the nervous system and that they were separate from each other. So he used Golgi's staining system, but he did this with infant brains, and so those separations for the synapses are farther apart. So he showed that the neurons did not grow into each other or fuse into each other as previously believed. Okay, so I'm going to talk about the two major categories of cells, the neurons and the glia. And I'll start here with the glia. I'm going to talk more about the different kinds of glia and what they do in this class than I do in any of my other classes. Glia is Greek for glue, so it literally means glue. And we used to just think of it as this kind of structural support that acted to kind of glue together the neurons. We know that they also supply nourishment to neurons, they remove waste material, and they also, so a special type of glial cell, make up those myelin sheaths that we're going to talk about. Those myelin sheaths are what provide insulation around many of the axons in the nervous system. I have these estimates at the bottom, but different people have different estimates, and I'm not really sure these are correct, but some people estimate that we have about 10 glial cells for every neuron in the cerebral cortex, and that they make up over 50% of the brain's volume. I often also do my little bits of nutrition as I'm talking about the nervous system, and if you want to keep your glia healthy, it looks like it's a good idea to eat your fish, get your fish oil, whether if you're not eating fish, and especially those smaller fish like mackerel and sardines, anchovies, that you are taking an omega-3, because really glial cells are made up of fats, and we, and especially in an American diet, we tend to get a bad ratio of the sort of omega-6s and 9s to omega-3s. We really need to up our omega-3s to keep our brain healthy. The other major category of cells is the neurons, and there are, again, many different kinds of neurons, which I will briefly talk about early on here, just so you're exposed to them, as sometimes some of these papers are looking at a particular kind of neuron. They're kind of interesting. And the neurons are really what are the cells that are responsible for communication, for the most part. So there's some discovery that some glial cells help with the synchronization of neural communication, so they are helping with communication, but for the most part, it's the neurons. They are doing the receiving, integrating, and transmitting of information. They're made up of a number of components here. So the first one is the soma. There's that Greek word for body, again, and this is the cell body. It contains the nucleus of the cell and the chemical machinery, or most of the chemical machinery. It has the pieces and parts that keep the cell alive. Branching off of that are the dendrites, and so they're going to branch out and receive signals from other neurons. And then we also have the axons. So every neuron has its axon that's going to transmit information away and communicate to other neurons. The axons are filled with a fluid that allow for this conduction of electrical signals. So we're going to see this electrical conduction going down the axon, and then we're going to communicate with other neurons by dropping chemicals with a chemical communication. There are three more general types of neurons: interneurons, which are wholly within the nervous system and communicating with other nerves or other nerve cells. Sensory neurons, so when we talk about sensation and perception, we're going to talk about these different sensory neurons that are receiving signals from outside the nervous system and taking those signals to the nervous system. And then motor neurons, so neurons that are carrying messages from the nervous system to the muscles. Like other cells in the body, neurons contain the following structures. So we have a membrane, and keeping the pieces and parts of the neuron inside the neuron, the nucleus, the mitochondria, the ribosomes, and the endoplasmic reticulum. And usually I point around here a bit, but I have slides with the verbiage on them, so I'm just going to go ahead and move to those. Okay, so the membrane, the structure that separates the inside of the cell from the outside environment. So basically keeping what's inside the cell separate from that extracellular fluid. You can see that it's made up of phospholipid molecules, so again, a kind of fatty substance, and that there are protein molecules, which we sometimes talk about as protein channels within that membrane. And as we talk about the membrane of the axon, it will be the first thing we start talking about, becoming selectively permeable to sodium or potassium. That is these protein molecules opening up to allow in that particular ion or allow out that particular ion. So we have protein molecules that allow for the transport of ions, sodium, potassium, chloride, and calcium, depending on a number of things. And again, we'll talk quite a bit about whether they are ligand gated, so when we have a neurotransmitter that affects these protein channels, which is the chemical or the ligand, or if they're voltage gated, so if it's the voltage of the cell that is influencing whether or not these protein channels open or shut. So we're in the soma of our cell, and within that we have the nucleus, the structure that contains the chromosomes, so our DNA, that hereditary influence on our cells, and the mitochondria, which is just plural for mitochondrion, those structures that are performing the metabolic activities and also doing a lot of the changing over of glucose to ATP, adenosine triphosphate. ATP is the energy, provides cells their energy for all their activities. Also here we have ribosomes, where we are going to synthesize new protein molecules. So we know that everything in our body is basically made up of proteins. So there's another healthy, nutritious tip, is to make sure you eat enough of your protein, as that is, especially if you are hurt in any way, as you're going to want to create those proteins, but we need them also, proteins, for our brain. And the endoplasmic reticulum, which you can kind of think of as a transport system, which is transporting those newly synthesized proteins to wherever we need them to go. And so neurons are cells, and they are very similar to the other cells of our body, except we do have different kinds of neurons, and those different kinds of neurons have their own distinctive shape, which really make a lot of sense for whatever function they are performing. So they vary in size, shape, and function, and we'll see that their function is influencing their shape. So this picture is showing a Purkinje cell of the cerebellum. And so these Purkinje cells, you can see that they have lots of dendrites and dendritic spines kind of branching off of that soma, because they are receiving information from many, many cells. And then they're going to, these Purkinje cells communicate to the kind of sub part, the lower part of the cerebellum. And so it's going to have this long axon coming off. It's because of this function of taking information from many, many cells and communicating it to another part of the brain that we get this particular size and shape. So here are some examples of different kinds of neurons. And there in A is, again, the Purkinje cell, which I just talked about, the Purkinje cell of the cerebellum. But we have Purkinje cells in other areas of the brain. So each of these examples is a particular example, but we might see that same kind of cell in other areas. So in B, we see what's called a unipolar neuron, as it has one pole coming off of the neuron. It is receiving information, in this example, sensory information from the skin, and then bringing it to the spinal cord. In C, we're looking at a pyramidal cell, as they are called, and this one is a pyramidal cell in the motor area of the cortex. In D, we have a bipolar cell, so two poles coming off that we might see in the retina of the eye, which they actually called just bipolar cells there. And then in E, he shows us the Kinyon cell of a honeybee, and you can just see the wide varieties of structures. I mean, this is just kind of interesting and neat to see how very different their cells are because of the really different functions. So I'm going to go through specifically the motor neurons and the sensory neurons and what they might look like. So this motor neuron has its soma in the spinal cord. It's going to receive excitation from other neurons, and then it's going to communicate to a muscle. So it's going to conduct impulses along that axon so that it can release acetylcholine to have the muscle contract. And here is another example. So that unipolar neuron that is really communicating from the skin to the spinal cord. In this example, although sensory neurons might have different appearances to them, they are really a specialized kind of neuron. And at one end, they are specialized to take in a particular type of stimulation. So in this case, in this example, it would be the touch of the skin, or temporal information about timing as some kind of vibrations on the skin. But depending on our sensory modality, they're going to be specialized to take in light, sound, chemicals, so forth. So most of this we've seen before. All neurons have the following major components, and we've talked about the dendrites branching off receiving signals, the soma, the cell body, which is the machinery of the cell, and then the axon, taking that information to neurons down the line. And because I'm taking this information, so we have a single axon coming off, but I'm going to communicate potentially with hundreds of neurons or thousands of neurons. And so to communicate with all those neurons, I'm going to have to, again, have these kind of branches coming off of the end of the axon, or sometimes it's more towards the middle, but the axon is going to have these presynaptic terminals, which allow for that neural communication, where we see the synaptic vesicles, we're going to see these little sacs filled with neurotransmitter so that we can communicate to other neurons. So the dendrites are branching out. They're going to receive the information, and their surfaces are lined with these synaptic receptors that are going to help us to bring information, take that information from that the neurotransmitters are telling us. Our neurotransmitters are going to fit into those receptors so that we can take information into the receiving neuron. The greater the surface area we have for these dendrites, the more receptors we can have, the more information we can receive. So some dendrites also contain, this is showing some little spines coming off of each of those dendrites. They branch out further, giving us more surface area so that we can have more synaptic receptors. And here's my basic picture that I usually talk from quite a bit. So you see the touch receptor and its soma, and that it's communicating to another neuron. Then it has its soma, and as I said before, for the somas, they contain all of that, the nucleus, the mitochondria. So we have mitochondria in the axon as well, but really the machinery for giving the cell its energy. There's protein synthesis in the ribosomes, and all of those other structures that we see in the cell body. And finally, we get to talk about the axons. When we discuss conduction of information to a new neuron, we are seeing this nerve impulse travel down the axon so that it can then communicate with other neurons or organs or muscles. A lot of axons are covered with this myelin sheath. As I mentioned in my specialized type of glial cell there, is made up of oligodendrocytes, which we'll start talking about. This is an insulating material. So if you imagine your kind of the wires of TV or anything, right, we insulate that with this kind of plastic insulation to keep that safe, but also it helps with efficiency, and it keeps the conduction separate from other neurons in this case as well. That myelin sheath has interruptions called nodes of Ranvier, which are going to become important when we start talking about that neural conduction. And then at the end of the axons, like I said, we have these presynaptic terminals that are the end points where we're going to release the chemicals for whatever neurons that we are communicating to. Okay, so some terms that are used to describe neurons, and sometimes so interneurons are sometimes referred to as just neurons that are completely within the nervous system. Afferent axons or afferent neurons are bringing in information. So sensory neurons have afferent axons, and efferent axons or efferent neurons are taking information out of the nervous system. So motor neurons are efferent axons. But sometimes this is talking about from kind of if you will, the perspective of a particular structure. So if we're talking about being within, let's just say, I'm going to use the thalamus, we're going to have afferent axons bringing information into the thalamus from the sensory neurons communicating to other neurons within the nervous system, but we're going to have afferent axons coming into the thalamus. If it helps you to remember, you can think of afferent axons or afferent neurons as arriving, as they both start with A. I don't know if these help, but hopefully they do. And then we have information leaving the thalamus, and it's going to communicate to some of the cortex, right? Those higher level processing areas. Those axons are going to be efferent axons. They're exiting, so those both start with E, if that helps you to remember those. And then interneurons, which again we can be saying just they're within the nervous system. And I think when I'm talking about a particular structure, it feels like I use intrinsic neurons. Some people use those interchangeably. But again, when we are communicating about staying within a single structure, interneurons or intrinsic neurons are those whose everything, the cell body, the dendrites and axons, everything of that neuron is within that particular structure. And we do have in the thalamus some neurons that communicate with other neurons that are still within the thalamus. Glia, our other major component, our other piece of hardware here, the major cells of the nervous system. I'm going to go through some of the types of glia and then show a picture that I think helps. I like the pictures as they kind of solidify things in my mind. The microglia are the ones that are some of the glia that remove waste and other microorganisms that could be harmful to the neurons. We can think of the microglia as being kind of the immune system of the brain. Another type of glia are the astrocytes. If you notice the beginning of that word astrocytes, astro, Greek for star or outer space, they are shaped kind of star-like. They do a number of things. I usually do this. What don't astrocytes do? They do everything. They do they do a lot. They provide the structural matrix holding neurons in place. This is one of the reasons glia have that name of kind of glue. They provide this matrix. They supply glucose, so nourishment to neurons by metabolizing nutrients from capillaries. Part of their relationship with capillaries is related to their role in the blood-brain barrier. So when I talk about the blood-brain barrier, I will mention astrocytes again. They clean up debris, so they remove waste. And they also allow for the isolation of a synapse. So if I am a neuron receiving information from several other neurons, there's all this extracellular fluid and ions kind of floating around, and we don't want all of that influencing our neural communication. So the astrocytes really protect that synapse from just floating ions and so forth from the extracellular fluid. Another thing that astrocytes do, so this is kind of more recent research as we talk about neurons being responsible for communication, but astrocytes are helping. They help to synchronize the activity of functionally related axons. They wrap around those presynaptic terminals and they can take up chemicals that are released by the axon and basically hold those chemicals until the other axons release their chemicals. And so those chemicals get released kind of at once, synchronously, which if you remember from our discussions of consciousness, that this is important for conscious experience and other aspects of the brain where we have these kind of really functionally related neurons that are trying to do a particular job. It's always a good idea to discuss oligodendrocytes and Schwann cells kind of in the same at the same time, but to also really distinguish them in our heads. So I've talked now about the myelin sheath and that kind of insulation that we see around many of our axons, not all of them, but around many of them. And the oligodendrocytes are doing this in the brain and the spinal cord. The Schwann cells are doing a very similar kind of job, building a myelin sheath in the peripheral nervous system. And they do this really differently. So for the oligodendrocytes, I'm going to have this body part of me, and it's going to send out these kind of offshoots. So if you see in the A part of the picture there, that those glial cells are wrapping around several different axons. And if I remember correctly, sometimes an oligodendrocyte can be influencing something like 50 neurons. So it's going to be sending these out quite a bit to wrap around and create that myelin sheath around those axons. In the peripheral nervous system, what the Schwann cells are doing is there's a Schwann cell, and it's going to just wrap itself around and around a particular area of an axon in the peripheral nervous system. One thing that this gives us is that the Schwann cells aid in the digestion of the dead and the dying axons and then arrange themselves. So they're already kind of, because of the way axons work, they're in these kind of cylinders, and they can line up in such a way that they guide the regrowth of damaged axons. And this is why in the peripheral nervous system we can see nerves regrow, right, and kind of fix themselves. Whereas in the brain and spinal cord, that's not what those oligodendrocytes are doing, and actually with other glial cells, they can actually be blocking the regrowth of the axon, so kind of hurting the ability for the brain and spinal cord to overcome this kind of damage. They also, and can't remember if it's oligodendrocytes, I think it's other glia that release that have this that release something that stops the growth. We'll talk about this in brain development and brain plasticity, that just causes the stopping of the growth of the axon, and then it'll go ahead and send out presynaptic terminals. But if it was communicating to a whole other part of the brain, it's not going to get back to where it was. And radial glia, we won't talk about too much, but again, just during neural development, they guide the migration of neurons and the growth of their axons and dendrites during that embryonic development. So really during brain development, and when that development is finished, radial glia differentiate into different things. So neurons, some into astrocytes or oligodendrocytes. I've been told to make my slideshows about 20 minutes long, and I thought, well, then I have some. I try to get them right around 23, 24 minutes. I'm a little bit over. I'll try to be a little bit under with the next one that comes up. But here's just again, the pictures that I find, excuse me, help solidify some of that. So on the left, we see the radial glia and this kind of migrating neuron, that it's guiding that neuron as we move to the right from there, and I'm going to go up, that astrocyte that has a kind of star-like looking appearance, and they are connected. They'll have these little feet that go along a capillary. Remember, we can take nourishment from those capillaries, but what this one is showing is part of what the astrocyte is doing with the blood-brain barrier. And then now I'm going to move down from that a little bit to the right again, as those oligodendrocytes sending out those paddles to create myelin sheath around several axons. And then moving farther to the right and up there, the Schwann cell. So that's one cell that the whole cell is wrapping itself around that peripheral nervous system axon. And then to the far right, the microglia. So the brain's immune system, these can kind of migrate around a bit so that they allow for the removal of waste, especially anything that's harmful to the brain.